Hello and welcome to Jeongbin's study room. Today, we are going to talk about spatial transcriptomics. Spatial transcriptomics began to gain the spotlight when it was selected as the Technology of the Year by Nature Methods in 2020. Spatial transcriptomics is a technology that analyzes RNA expression while maintaining the positional information of tissues or cells. Traditional single-cell RNA analysis methods, which analyze RNA at the single-cell level, have limitations in that they cannot capture actual cell-to-cell interactions that occur only when cells are spatially adjacent. To overcome this limitation, methods for analyzing RNA within spatial contexts have been developed, and one of these methods is spatial transcriptomics. This technology allows us to determine the location of different types of cells, and it has advanced to the point where it is possible to analyze RNA expression at a subcellular level, even showing where RNA is located within individual cells. For example, in cancer tissues, even cells of the same cancer type can show different gene expression depending on their interactions with surrounding cells. Spatial transcriptomics allows us to understand these interactions and the microenvironment. It enables the analysis of spatially significant tissues, such as the different layers of the brain, like the frontal, occipital, and temporal lobes, or the layers of the skin, revealing distinct characteristics within each layer of an organ. In short, spatial transcriptomics enables new analyses by combining it with other spatial biological data, adding a new dimension, space, and providing new information. Similar signals obtained from cells are often spatially adjacent, so spatial proximity itself provides statistical significance. Spatial transcriptomics provides transcriptomic data in a spatial context, offering crucial insights into cell-to-cell interactions and tissue structure. It allows us to observe interactions between spatially adjacent cells and examine the characteristics that emerge at different spatial scales, making it possible to study biological emergent properties. One example of spatial transcriptomics is 10x Genomics' Visium. In this technology, each spot has a spot-specific oligonucleotide. These oligonucleotides hybridize with RNA molecules derived from tissues, allowing the acquisition of transcriptomic data for each spot. This enables the mapping of gene expression across the entire tissue with high spatial resolution. The 10x Visium technology works as follows. First, a tissue section is placed on a slide coated with oligonucleotides. As the tissue permeabilizes, RNA molecules are released and bind to capture probes in each spot. Once bound, reverse transcription occurs to create cDNA, which is then sequenced. The sequenced data is mapped back to the spots, allowing the spatial interpretation of gene expression profiles across the tissue. In addition to the standard 10x Visium, there are also probe-based Visium methods that target specific gene expressions, enhancing the specificity and sensitivity of the analysis. Another notable method is slide-seq. In slide-seq, tissue sections are placed on a surface coated with microsphere beads containing barcodes. These beads capture RNA, and the unique barcode of each bead helps spatially position the transcriptomic data. Like 10x Visium, this also generates high-resolution gene expression maps. Both Visium and slide-seq are in situ-based technologies. In addition, there are other types of spatial transcriptomic technologies, such as image-based and region of interest-based approaches. A representative example of image-based technology is Xenium and Merscope. Xenium allows for the immediate imaging of highly detailed RNA and protein information, offering fast integrated analysis, while Merscope provides high-resolution visualization and quantification of RNA transcripts within tissues. With spatial transcriptomics technology surpassing the limitations of existing methods, it is expected to play an essential role in personalized medicine, drug development, and personalized therapies. It has provided a new paradigm for understanding cells and tissues. In which fields do you think spatial transcriptomics will play the most important role in the next 10 years? Leave your thoughts in the comments. I'll be waiting for you at Jeongbin study room.